We are going to break Chapter 5 into two separate units. For this section, we will only be looking at the first topic in Chapter 5, the plasma membrane. This will cover sections 5.1 to 5.9. Biologists use the term fluid mosaic model to describe the structure of the plasma membrane. Let's break that term down. Fluid is something liquid, and it usually has movement. This refers to the phospholipid bilayer that makes up the plasma membrane. It has a fluid consistency, like that of an oil-based salad dressing. A mosaic is something made of many pieces that come together to make a bigger picture. Think of taking broken tiles of many colors and arranging them into an image. The mosaic of the cell refers to all of the proteins in the membrane. They each have an individual function that contributes to the overall function of the membrane. There are many different types of proteins that can be found in the membrane, and they have a huge variety of functions. Membrane proteins can be used for attachment, adhering the cell to other cells. Receptors receive signals from other cells. Transport channels allow some molecules to pass through without needing energy, while active transport proteins require energy to move molecules across the membrane. We're going to focus on these last two functions in the next parts. The plasma membrane is also called selectively permeable. The word selective refers to the ability to choose. The membrane will let something pass through, but not everything. It's selective. The word permeate means to spread out or pass through. So a selectively permeable membrane will allow some substances to pass through it, but not others. Phospholipids are the main component of the plasma membrane. Their specific properties allow them to form membranes spontaneously. Spontaneous just means it happens without needing any energy. They have a polar head, which likes to associate with water, and a nonpolar tail, which doesn't like to associate with water. This means that when they are put in water, the heads will point out towards the water and sandwich the nonpolar tails in between them. Watch the linked video to see the actual molecules arrange themselves into a bilayer membrane. The spontaneous formation of membranes was likely the first step in the evolution of the first cells. This made it possible to distinguish between the inside and the outside of a cell, and they were able to start developing replicating molecules inside of the cells.